The very beautiful thing about the CAD is it can be incorporated at any stage of the drug discovery. What does that mean? So, as I told you, from the target selection to the launch of the drug, it is a multi-step process. For example, you want to check you want to screen 1,000 compounds against one target. It is not possible manually, like it can be done, but it will take a lot of time. Okay, so now you want to do that, the screening of these 1,000 compounds against one single target, that you can do computationally, and rest of the thing you don't want to do computationally, that is okay, leave it. You just do the screening, computational screening, and then rest of the thing you can do experimental. Okay, for example, you have done the screening, you have already done the screening with the experimental method. Now, the further thing, the optimization of the compounds, the evaluation of the compound, that you can do with the help of computation. So this can be incorporated at any stage. It is not necessary like if in the beginning you have to start with the computational methods, only then you can go for the CAD. No. If you have started the drug designing process and at whatever stage you are, you can incorporate the CAD methodology. And what is this CAD methodology? Nothing but the available resources and methods, the computational methods. That's it. It is a multidisciplinary, of course, as we are talking about the drug design, so many things will be there. Okay. And it has very few side effects. In fact, no side effect. And if we talk about the few side effect is sometimes what happen, as we are working on the computational method, all these are computational methods or algorithms are designed by man only, right? These are also man-made. Some mistake can happen. But if you are expertise, if you are in the, under the surveillance of any expertise, then this side effects is not there. Now comes the multidisciplinary approach. That means if we are talking about the drug designing, then we have to make sure like this similarity search, target identification, structure prediction. What are these? These are the methods. These are the steps when we work in the CAD. Okay. Similarity search means if we have one single compound, we can go for the similar compound of that particular. Okay. That is the similarity search. And what you have to do? You have to use the available resources for that. You don't have to do it manually. Then the target identification. Lot of literature publications are available. From there, you can easily understand or easily identify the target. If you look here in this figure, you will see what are these. These are the small compounds. Just assume these are 1,000, 10,000 compounds are there. And this one single target protein is there. The target protein, which is responsible for the pathogenicity of the disease, ABC, whatever. Now, what we have done, when we are checking the binding of these ligands or these small compounds, you can call them ligands as well, of these small compounds to this protein, that is the procedure of screening. Okay, once the screening is done and we have identified few compounds which are actually binding to the active site of the protein. Now the question is, what is the active site? So basically, in a protein is a set of like the amino acids, many amino acids will be there. Okay, out of those amino acids, 2, 3 or 1 or 2 amino acids are those which are responsible for the activity of the protein. And what we have to work on, we have to work on the activity of the protein, right? Of course, for example, the protein A is causing the disease B. If that, what is the activity of that protein A? That helps in the causing of the disease, right? So we have to stop that activity. And how will we able to stop the activity? If we will be able to stop the function or performance of the active site residues. That is our aim. Okay. So it is binding to the active site residues. Once it is binding, we have to further go for the multiple studies. Like we have to check whatever the compounds we have identified, whether it is safe or not. Okay, multiple properties we need to check. Further, we have to check whether it remain bound or not. If sometimes what happens, the compound is binding, but after some time it is drifting away from the protein. That is also good, not a good thing. So further it comes the multiple things. So after that, out of these compounds, only 2 or 3 will reach at final stage. Okay, final stage before clinical trials. And few of them might fail at the clinical trial stage, and only 1 or 2, hardly 1 or 2 compounds will become the drug that will be launched into the market. One more thing, CAD is completely dependent on the experiment. Like whatever you will be doing, whatever compound you will be identifying using the CAD methodology, that need to be validated experimentally. Okay, only then you will be able to further process in the clinical trials or market launch. That is a brief about the CAD. All these things we are going to understand in the deep. Okay. First of all, let's talk about the CAD objectives. Why the CAD has been introduced. If the drug, of course, the drug designing was going on before CAD era also, right? Why the CAD has been introduced? Basically, the very important reason is the to speed up the screening process. As I told you, we are dealing with thousands of compounds. It is not easy na, to screen each and every compound experimentally. You have to buy the compound, you have to prepare the assay, and so many things you have to do. That is the time taking process. So that screening time will be reduced using the CAD. It is very focused and target directed screening. Like one target will be there, and us, whatever the active site residues are there, we will be focused on only those. We don't care whatever other things are happening to the protein, what other things are, compounds are doing with the protein. We are not interested in that, as we are interested in the CAD only. Okay.